Usually when we think about our DNA, we just kind of picture it as this fixed master blueprint. Like you have a gene, it gets turned on,
Yeah.
and then it builds a very specific molecular machine for your body. It's a very comforting, simple picture.
Right, yeah. It implies this very binary world, like the gene is either expressing, meaning the machine is being built, or it's not.
But what if I told you that knowing a gene is turned on is actually only half the story? What if that exact same gene can produce entirely different molecular machines, like entirely different tools, depending purely on where that cell physically sits in your tissue?
I mean, it completely shatters that simple blueprint metaphor. We are actually looking at a layer of biological complexity that is highly dynamic and heavily influenced by geography. And honestly, historically, it's been incredibly difficult to measure.
And that brings us to our deep dive for today. We have this really fascinating stack of research in front of us, anchored by a groundbreaking January 2026 paper from Nature Biotechnology. It's titled Mapping Isoforms and Regulatory Mechanisms from Spatial Transcriptomics Data with SPLISOSM.
Yeah, the authors here, they're led by researchers at Columbia University, have essentially given us a completely new mathematical lens. They've built this algorithmic engine that lets us see this hidden layer of spatial biology in its native habitat. And the cool part is they do it without destroying the data in the process.
So our mission today is to exhaustively unpack this paper top to bottom for you. We're going to decode the biology of what we call isoforms. We'll explore the massive mathematical hurdles that have kept this hidden from us. And we'll look at how this new algorithm, SPLISOSM, is fundamentally changing how we understand both healthy brain function and devastating cancers like glioblastoma. Okay, let's unpack this.
So to really grasp what this paper achieves, we first have to ground ourselves in the biological foundation, which is transcript diversity. I mean, almost all human genes undergo this process called pre-mRNA processing.
Okay, well, let's visualize that mechanism for a second. So you have your DNA, which gets transcribed into a messenger RNA, or mRNA. But it doesn't just, roll off the assembly line and go straight out to build a protein. It gets edited first.
Exactly. The cell essentially cuts and pastes that messenger RNA sequence before it is finalized. And there are two main ways this happens. The first is alternative splicing.
Right.
This is where the cell cuts out certain middle sections of the RNA, we call those introns, and pastes the remaining parts together in different combinations. It's like editing the middle scenes of a movie to change the plot.
So I like that.
And the second way is alternative 3' end usage. This is where the cell decides where to literally cut off the tail end of the RNA. It's essentially choosing where the movie's end credits roll.
So the result is that a single gene can create multiple distinct versions of its transcript. And these different versions are the isoforms, right?
Yeah, exactly.
So think of a gene as a master recipe for a cake. Splicing is like handing that exact same recipe to different localized bakers. The baker in one neighborhood might decide to swap walnuts for pecans or change the vanilla frosting to chocolate, just based purely on local tastes.
That's a great way to put it.
The master recipe, the gene, is exactly the same, but the resulting cake, the isoform, is totally customized for that specific neighborhood.
And those localized tastes, the bakers in your analogy, those are driven by RNA binding proteins, or RBPs.
Okay.
These are physical molecules that bind to the RNA and dictate exactly how it gets sliced, diced, and capped. And what's really crucial here is that the human brain exhibits incredibly high transcript diversity. This isn't just, random editing noise.
Right, it's heavily regulated machinery.
Yeah.
The brain actively relies on these customized cakes to function.
Yes. The localized expression of these RNA binding proteins drives very specialized transcript landscapes. This location-based editing is fundamental for neurogenesis, which is the birth and differentiation of new neurons.
Wow.
And it's also vital for synaptic plasticity, which is how our neurons change how they communicate so we can actually learn and form memories.
I imagine when that delicate balance is disrupted, like when the local baker makes a mistake and starts producing the wrong isoform for that specific brain region, the clinical implications have to be severe.
Oh, absolutely. Disruptions in this splicing balance are deeply linked to numerous neurological disorders. And beyond the brain, we also see this exact system hijacked in cancer progression. Tumors will use this location-based editing to physically adapt to different immune environments within the body.
Okay, let's pause and make sure we have this first foundation locked in. Let's do a quick 5-minute bullet recap. Number one, genes aren't fixed blueprints. They make multiple customized versions called isoforms via splicing. Number two, the brain relies heavily on this transcript diversity for complex functions like memory.
All right.
Number three, this is all regulated by local bakers called RNA binding proteins, or RBPs. And number 4, when this location-based editing breaks down, it leads to severe neurological disease and cancer.
Spot on.
Think about your own brain for a second. What really stands out to you about the idea that the physical location of a neuron literally dictates how it reads its own DNA? It's mind-bending. But hey, if this location-based editing is so critical, why are we only just now mapping it out in 2026?
Well, the limitation has always been the technology, or more specifically, the massive, massive data problem that the tech creates. The field we are talking about here is spatial transcriptomics, or ST.
Right, and ST platforms are the tools that let us see not just what genes are active, but where they are on a physical map of the tissue.
Exactly. Technologies like 10x Visium or SlideCQV2, they do this by laying down a microscopic grid of barcodes on a glass slide. You place a thin slice of tissue on that grid, and the RNA from the cells binds to the barcodes.
So it tags them.
Right, so when we sequence that RNA later, the barcode tells us the exact X and Y coordinates where that specific molecule originated. And newer imaging-based platforms, like Xenium, they do something very similar but with fluorescent tags.
So these platforms naturally capture the isoform-specific information. We literally have the data in front of us. But historically, most analysis pipelines just clumped all that rich, specific transcript data together at the broad gene level. They literally threw away the isoform details. Why would researchers throw away the good stuff?
Because the math required to process it is a complete nightmare. Older algorithms simply couldn't handle it. There are 3 major mathematical bottlenecks when dealing with spatial isoform data.
Okay, to help us remember this, let's use the mnemonic SMS. SMS, what's the first S?
The first S is sparsity. Spatial transcriptomics data is notoriously sparse.
Meaning it's mostly zeros, right?
Yes, mostly zeros. When you take the total counts of a gene and then try to subdivide them across multiple different isoforms at thousands of microscopic locations, you inevitably hit 0 counts at many of those spots. You simply didn't capture enough molecules to measure the difference between isoform A and isoform B.
Okay, so it's like trying to calculate the ratio of chocolate versus vanilla cakes sold on every single street corner in a whole city. On most tiny street corners, 0 cakes were sold, so you literally can't calculate a ratio at all.
Exactly. And if you carry that logic forward, previous workarounds involved adding what we call pseudocounts.
Pseudocounts.
Yeah, basically injecting a small arbitrary number like 0.1 into those 0 spots just to avoid having the computer divide by 0 and crash. But doing that introduces massive mathematical biases that completely distort the true biological relationship.
Wow. Okay, so that's the sparsity bottleneck. Now, what about the M in our SMS mnemonic?
The M stands for multivariate and compositional. Isoforms from the same gene are mathematically interdependent. If a gene produces 100 total transcripts and 60 are isoform A, then exactly 40 must be isoform B. Their relative ratios must sum to 1. They are a composition.
Like slices of a pie. If one slice gets bigger, the other slices mathematically have to get smaller.
Yes, and if you ignore that mathematical reality, which frankly many older statistical approaches did, you end up treating those pie slices as independent variables. You end up thinking the whole pie is growing when it really isn't.
Okay, so S was sparsity, M is multivariate. What's the final S?
The final S is spatial autocorrelation.
Ah, the geography rule.
Right. Things close together are naturally more similar than things far apart. In standard statistics, most differential association tests assume your observations are completely independent, like flipping a coin. But in spatial data, nearby cells influence each other. A cell is going to act a lot like its neighbor.
So if you ignore that,
If you ignore spatial autocorrelation, you generate massive false positives. You'll see 2 completely unrelated genes varying together and assume they are interacting, when really they just both happen to be reacting to the same local geography. It's just structured noise.
wait, I need to push back and really understand the scale of this failure. The paper mentions previous methods like SparkX or tools using basic log ratio transformations. Were they basically flying blind or like creating hallucinations in the data?
What's fascinating here is that they were absolutely creating hallucinations. Early attempts either inappropriately treated these interdependent pie slices as completely independent, or they relied on oversimplified metrics that couldn't handle the geography rule.
So they broke their own math.
They fundamentally destroyed their own statistical power. They were looking at a highly complex, interdependent spatial network and essentially blurring the image until they saw what they wanted to see.
Man, let's do a quick synthesis of this data problem before we look at the solution. Here's our second 5-minute bullet recap. Number 1, ST platforms capture spatial data, but older tech clumps it together. Number 2, the SMS problem ruins standard math. S stands for sparsity, too many zeros.
Yep.
M stands for multivariate isoform ratios are pi slices that must sum to 1. And the last S stands for spatial autocorrelation data. Nearby cells aren't independent coin flips.
Exactly.
Imagine trying to map a major city's traffic patterns using blurry, interdependent snapshots where half the cars are just missing. How would you even solve for that missing data without making terrible assumptions?
Well, you would need a completely new algorithmic engine, and that is exactly what the Columbia team built. It's called SPLISOSM, Spatial Isoform Statistical Modeling.
Okay, let's look under the hood of SPLISOSM. How does it actually solve these impossible bottlenecks?
It completely reformulates the math from the ground up. Instead of using standard linear models, it casts spatial variability as a series of multivariate independence tests. And the core engine running this is something called HSIC, the Hilbert-Schmidt Independence Criterion.
Okay, HSIC. Explain the mechanism of that to us. What is it actually doing to the data?
Think of it as a way to find hidden nonlinear relationships. HSIC uses what are called nonparametric kernels. A kernel is just a mathematical function that takes your data, in this case, the isoform ratios and the physical XY spatial coordinates, and projects them into a highly complex, multidimensional feature space.
Okay, so if the pattern of where these isoforms live is completely invisible to us on a flat 2D map, HSA throws that data into 3D or 4D, twisting it around until the shape of the relationship suddenly becomes obvious.
That's the perfect visual equivalent. But SPASism introduces two major technical innovations to make HSIC work for our specific spatial transcriptomics problems. The first is outlined in their Theorem 1, which tackles the spatial kernel. They mathematically prove that low-rank approximations inevitably sacrifice statistical power.
Wait, low-rank approximations being the mathematical shortcut that older tools like SparkX use to process data faster, right?
Yeah, right. To process thousands of cells quickly, older tools essentially compress the spatial data. But to avoid losing power, SPLISOSM absolutely refuses to take that shortcut. It uses a full-rank intrinsic conditional autoregressive model, or ICAR, which is based on spectral graph theory.
Let me try to break down how that preserves the data. Older algorithms were compressing the image to save processing time. It's like taking a highly detailed satellite photo of a city and blurring it into 100 big neighborhood squares just to save memory. It's fast, but you lose the individual houses.
Yeah, you lose all the detail.
But SPLISOSM's ICAR model looks at every single house on the street. It maps the exact relationship between every single adjacent spot without compressing it, keeping all those high-frequency, sudden local changes completely intact.
And that granular mapping is exactly why it doesn't hallucinate false positives. Then we have their second innovation, Theorem 2, which tackles the sparsity problem, the compositional kernel.
Wait, I need to push back on this. You just said this data is incredibly sparse, meaning mostly zeros. If they refuse to use pseudocounts to fill in the gaps with a fake 0.1, how on earth is SPLISOSM dividing by 0? You can't just magic math out of thin air to solve an empty street corner.
You're right, you can't. Which is why they developed a new kernel that uses a mean replacement strategy. Instead of injecting a fake 0.1 into a single blank spot, the kernel matrix looks at the average ratio across the entire tissue. It uses that global baseline to mathematically anchor the calculation for that specific zero count spot.
Oh, so it's essentially saying, hey, we lack data at this specific house. So rather than making up a fake local number, we will just assume it matches the global city average just so the matrix doesn't collapse.
Exactly. It borrows statistical strength from all the spatial observations to maintain the validity of the overall test without ever inventing artificial local spikes. And with these kernels dialed in, SPLISOSM gives researchers 3 distinct spatial variability tests.
Which are?
There's HSIC-GC, which tests for spatial patterns in the total gene count. There's HSIC-IR, which looks purely at the isoform usage ratio, the pi slices, and HSIC-IC, which looks at the raw isoform count.
And once it finds a gene that is actively varying its isoforms based on location, it goes a step further into causal discovery, right?
It does. It uses differential usage, or DU tests, to find the RNA binding proteins, the bakers making the changes. SPLISOSM uses conditional HSIC tests.
How does a conditional test find the baker?
By mathematically freezing the geography, it conditions the test on the spatial location. It essentially says, if we hold the physical neighborhood constant and ignore where we are, does the presence of this RBP still perfectly correlate with the change in the isoform?
Wow.
Right? If the answer is yes, you've eliminated the spatial autocorrelation noise. You've proven the protein is actively causing the edit, not just hanging out in the same neighborhood. If we connect this to the bigger picture, this allows for cell-type agnostic discovery.
Meaning you don't even need to sort or label the different types of cells on the slide to find the regulatory mechanisms. The math just finds the cause and effect directly.
It's an incredibly robust system.
So let's gather up this algorithmic deep dive for our third recap. Bullet 1. SPLISOSM uses HSIC to project data into higher dimensions to expose hidden patterns. Bullet 2. It rejects the fast, blurry shortcuts of older tools, using a full-rank ICAR model to map every tiny spatial relationship. Bullet 3. It solves the missing data problem by anchoring empty spots to a global average, rather than faking data. And Bullet 4. It uses conditional testing to freeze geography and prove exactly which proteins are causing the RNA edits.
Perfect summary.
Does the math make sense to you? It's basically a highly advanced filter separating true biological instructions from random geographical noise.
Which brings us to the ultimate proof. Having a sophisticated mathematical theory is one thing, but actually applying it to chaotic, living tissue is where the true value lies.
Here's where it gets really interesting. Let's look at the biological evidence. When they finally unleashed SPLISOSM on real tissue, what did they actually find?
Well, they started by establishing level 1 evidence using rigorous simulations. They generated massive artificial data sets where they knew exactly what was a true signal and what was noise. And SPLISOSM produced perfectly calibrated p-values.
And a p-value basically being a measure of statistical surprise, right? Like, how likely is it that this pattern happened by random chance?
Exactly. And usually in spatial data, calculating accurate p-values requires permutation testing, which means having a computer shuffle the data thousands of times to establish a baseline. It takes forever.
But
SPLISOSM's math calculates it directly and flawlessly. And crucially, its HSI-IR test was completely unfooled by total gene expression changes. It purely isolated the hidden isoform usage patterns.
So the algorithm passed the simulation stress test with flying colors. Then they aimed it at the adult mouse brain.
And that's where the scale of what we've been missing became apparent. SPLSRs discovered over 1,000 spatially variable transcript diversity events that previous methods had completely missed.
Over 1,000. We've essentially been looking at the brain with one eye closed this whole time. By just opening that second eye, the isoform data, we instantly discovered 1,000 new moving parts that were entirely invisible to older tools. Give me an example of one of these moving parts.
Sure, look at the genes MAP4 and QQ. SPLISOSM mapped out precisely how their specific isoforms change across different microscopic regions of the hippocampus and the cortex. These aren't just, random housekeeping genes. They're heavily involved in synaptic signaling pathways.
The exact pathways linked to major neuropsychiatric disorders.
Exactly. And through those conditional tests we discussed, the algorithm uncovered both known and completely novel regulatory relationships with region-specific RNA binding proteins. It didn't just find the altered cake, it found the actual local baker in the hippocampus responsible for the edits.
But mouse brains are highly structured, controlled environments. Does this spatial logic hold up in humans?
It really does. They analyzed spatial transcriptomics data from the human prefrontal cortex and found that these spatial splicing patterns are evolutionarily conserved. The exact same geographical editing rules applying to mouse brains apply to ours.
Okay, but then the researchers took SPLISOSM into what might be the most chaotic biological environment on Earth. Human glioblastoma,
Right.
brain cancer.
Yeah, solid tumors like glioblastoma are notoriously heterogeneous. The tissue is an absolute mess of mutating tumor cells, invading immune cells, and dying necrotic tissue.
Right.
Traditional transcriptomics methods require you to sort the cells into neat, defined types before analyzing them, which is incredibly difficult when a tumor is actively mutating its own identity.
But earlier we established that SPLISOSM is cell-type agnostic.
Exactly. Because SPLISOSM just looks at spatial dependence, it bypassed the messy cell typing step entirely. It just looked at the coordinates, and it revealed pervasive transcript diversity in genes responsible for antigen presentation and cellular adhesion.
Antigen presentation. Let's explain the stakes of that mechanism. That's essentially how the tumor shows its face to the immune system.
Yes,
Right?
it's how it signals what it is.
Yeah.
And SPLISOSM showed that the tumor is literally altering its RNA splicing, changing the isoforms of its immune-facing proteins based specifically on the microenvironmental conditions it's physically sitting in.
Wait, so the tumor is editing its own recipe book on the fly, literally changing its physical disguise depending on which specific neighborhood of the brain it has invaded. That is terrifying, but also incredible to be able to finally see mathematically.
It's totally wild.
Let's synthesize this biological evidence for our final recap. Bullet 1. Simulations prove the math works flawlessly without generating false positives. Bullet 2. In the mouse brain, opening the isoform eye revealed over 1,000 hidden splicing events.
All right.
Bullet 3. Those mechanisms are evolutionarily conserved in humans. And bullet 4. In glioblastoma, it proved tumors dynamically change their genetic disguise based purely on their immediate physical surroundings. If a tumor can change its gene isoforms just based on where it's sitting in the brain, how does that change how we think about treating it?
It really is a paradigm shift in how we analyze tissue architecture.
So what does this all mean for you? We are witnessing a monumental shift from a gene-centric view of biology to an isoform-centric view. Knowing that a gene is on is no longer enough.
Not at all.
If a tumor can change its gene isoforms, literally altering its physical structure just based on where it's sitting, a drug that targets one isoform might be completely useless against the other side in the exact same tumor. We now finally have the mathematical algorithms to see the actual biological machinery being custom-built in its native habitat.
This raises an important question, though. The data SPLISOSM needs to do this already exists in many cases.
Wait, really? We don't have to run all new experiments.
But the beauty of this field is that spatial transcriptomics platforms have been capturing this raw, detailed RNA data for years. But because the older algorithms couldn't process the sparsity and the spatial autocorrelation, that rich isoform data was quite literally discarded as noise. Researchers can now take SPLISOSM and apply it retroactively to thousands of existing data sets.
They can mine old data for entirely new biological discoveries. The blueprints have been sitting in the archives this whole time. We just finally have the right pair of mathematical glasses to read them.
It's an unprecedented goldmine waiting to be reanalyzed.
That is incredible. I want to leave you with a final thought to mull over. We opened by talking about how a single gene can be edited into entirely different machines depending on its physical location. If the precise location of a cell dictates how it edits its own genetic recipe, then our bodies aren't just chemical factories. They are incredibly complex, location-aware architectural maps.
Exactly.
Every single cell is constantly asking, where am I and what tool do I need to build right now? What other secrets are hiding in the spatial layout of our organs, waiting for the right algorithm to finally decode them?
The spatial architecture of life is just beginning to unfold.
Thank you for joining us on this deep dive. Keep questioning the blueprints and keep your curiosity sharp.